Hi, welcome back. In this video we are going to look at the organic polymers that are proteins. So let's get started straight away. What atoms do you find in proteins? Well, proteins contain carbon, hydrogen, nitrogen, and oxygen. And then two amino acids, cysteine and methionine, also contain sulfur. So proteins are these organic polymers. They have these building blocks, these monomers we've talked about before, and those monomers are called amino acids. So let's look at the structure of an amino acid. Here's a generic amino acid, and what I mean by generic is that it has the same, all amino acids have the same basic structural formula, and then there's a variation around what we call the R group. So we'll come back to the R group in a moment. Before we look at the R group, let's take a look at some of the other components of the amino acid. So all of the functional groups in an amino acid are arranged around a central carbon atom called the alpha carbon. So if you're asked to draw an amino acid, always start with the alpha carbon. Just put a C down in the middle of your page, and that's the alpha carbon. And then from there, knowing that the valence of carbon is 4, you've got to add 4 bonds somehow, and they're going to be single bonds. A bond that's made with the R group we'll come back to. There's a single hydrogen atom bonded to the carbon, the alpha carbon, and then on the left, as we've got it drawn here, we've got a functional group that contains nitrogen, and in this case 2 hydrogens, and we'll see a little variation of that in a moment. So this is a new type of functional group. This is an amino group, so you should be able to recognize this one, draw it, and talk about its chemical properties. So what are its chemical properties? Well, we've got a nitrogen, and that nitrogen is covalently bonded to 2 hydrogen atoms. Now if you think back to our discussion on electronegativity, hopefully you recall that nitrogen is much more electronegative than hydrogen. So what's going to happen here is that electrons will be drawn towards the nitrogen in this covalent bond, and that means the nitrogen is going to be slightly negatively charged and the hydrogens will be slightly positive charged. So we've got a polar functional group here, but that's actually not important when we look at the chemical properties of amino acids, and we'll talk about why that is when we get to the R group. So there's an amino functional group. Now the amino functional group can either have 2 hydrogens like you see here, or it can have a third hydrogen attached to it, in which case the nitrogen would have a positive charge on it, and so the end would have a little plus next to it, and we'll see an example of that in a moment. So down at the other end, attached to the alpha carbon on the right hand side, as we've got it drawn here, is our old friend the carboxylic acid group, and here I'm showing it as a C double bonded to an oxygen and then a C bonded to a hydroxyl group. So we've seen this before. Remember, it's a carboxylic acid group, so what that means is that at certain pHs, this hydrogen down in the bottom right here, that will actually leave and will end up leaving an electron behind from its shell, and so it will leave as a proton. And so remember, this is going to behave as an acid because it increases the concentration of protons in solution, and that suppresses the pH, which is what an acid does. So let's come back to this R group here. So when you look at the chemical properties of amino acids, we tend to ignore the contribution of the amino group and the contribution of the carboxylic acid group. And that's because of something we'll look at when we look at how these things polymerize, and you'll see why we ignore the amino group and the carboxylic acid group when we come to joining these monomers together. So what is this thing here, the R group? R in chemistry language, I guess, just means the rest of the molecule, the remainder of the molecule. And the R there is highly variable. There are 20 common amino acids, and the R varies. So the simplest amino acid is just a hydrogen, so the R group there is just replaced with a little hydrogen atom. And then there's a whole variety of different functional groups that can be added in place of R. We can have chains of functional groups. Now, I don't want you to go away and learn the 20 amino acids, but you have to understand and you have to know that the R is variable, and it's the chemical properties of the functional groups in the R group that gives us the overwhelming chemical property of the amino acid. So if you go back to thinking about the electronegativity of atoms, and I give you an R group, you should be able to work out the chemical properties of the R group based on your understanding of electronegativity, and then how that leads to polarity or non-polar groups. And from there you can start using the language of hydrophobic, hydrophilic, that kind of thing. So let's take a real quick question, just as a little bit of a pause here. Here's a question. At physiological pH, the COOH, that's the carboxylic acid group, of the amino acid donates a proton into solution, which increases the proton concentration, so suppresses the pH there. How would we classify the COOH group of the amino acid? So I just answered this, but this is the kind of the kind of question which requires that you apply a little bit of understanding from another part of the class. So if you've got a functional group and at a certain pH you're told that it loses a proton, that increases the proton concentration of the solution that thing's in, and that suppresses the pH, and that's what an acid does. So the carboxylic acid group is an acid by definition. But if I just told you COOH and then told you that it loses a proton, you've got to work out what's going on. So let's try a kind of variation of this one. Here's another question. I'll stop talking and let you read and answer, and I'll put the answer up in a little bit. Okay, so we've got some idea about the generic structure of an amino acid. Let's take a look at a specific example. Let's look at two examples. Here's aspartic acid, also known as aspartate. So let's see if we can identify the functional groups we just talked about in the generic amino acid structure. I've made it a little easy here because it's drawn in exactly the same orientation as what you just saw. Now remember, this could be flipped upside down, it could be drawn the other way around, all kinds of variations. So don't just get used to seeing these molecules in one orientation. You could be looking at this from the other side, flipped, rotated, upside down. So get used to identifying the alpha carbon to start with. So that's always this central carbon here. So there's the alpha carbon forming these 4 bonds. So we know that's the alpha carbon. Here's that hydrogen that hangs off the bottom as it was originally drawn. And then NH2 is just another way of showing the amino group. And now here's our carboxylic acid group. And so in red up here, that's our R group. And so we've got a CH2 here, and then we've got another carboxylic acid group here. So if we looked at this amino acid and they said at physiological pH, what's its characteristic going to be, you should be able to apply your understanding that at physiological pH, this carboxylic acid group, just like this one here, would dissociate. And so this is literally, as the name says, it's an acidic amino acid, and it's an acidic amino acid because of this carboxylic acid group here, not because of this one here. Let's take a look at another one. Now this one I've made a little bit more complicated. This is valine, and so here I've not drawn the amino acid like you expected it to be drawn. So I'm going to pause here for a moment and just let you look at this, and I want you to do a few things. First, just draw the amino acid down on your notepad, and then I want you to label the alpha carbon, the carboxylic acid group, the amino group, and then the R group. And the R group is going to be pretty easy. And once you've labeled the R group, I want you to work out what its primary here, what its actual chemical property is. So you're going to label the alpha carbon, the carboxylic acid group, the amino group, the R group, and then write down the chemical property of the R group. So I'm going to pause, stop the video, and I'll start it up again in a moment. Okay, so what have we got going on here? So here is our alpha carbon. Now it's drawn a little bit kind of funky here because the carbon only looks like it's making 1, 2, 3 bonds, but the chemist has written the H attached directly to the carbon, so you could put the H up the top here, you could put the H up here as well if you wanted to. So here is the alpha carbon, and then here's our NH2 group, again written a little bit differently, and then here's our carboxylic acid group. Now you could draw it flipped like this as well, and that would be completely okay, because cells don't really have left and right and up and down, they have inside and outside. We'll talk about that when we get to membranes. And so here's our alpha carbon, our amino group, our carboxylic acid group, and then the R group is easy to identify because it's shown here in orange. And if you're looking at that R group and asking yourself what is the chemical property of this amino acid, we're only looking at the R group, remember, and so what we've got is a lot of carbon-hydrogen bonds here. That's a carbon with 3 hydrogens on it, and there's another one there, and then a carbon bonded to a hydrogen. So electronegativity of carbon and hydrogen are about the same. So in those bonds between the carbon and the hydrogen, the electrons are being pulled equally. They've got an equal probability of being around the carbon and the hydrogen as far as we're concerned, and so those bonds are non-polar covalent bonds. And so this is a non-polar functional group. It's kind of oily, it's fatty, it's hydrophobic or non-polar. So that's a quick analysis of an amino acid. I can do that for lots of different amino acids, so expect to see some of those on quizzes. Okay, so let's now get into the business of joining some amino acids together. But before we do that, we've got a little bit of a variation on how these amino acids have been drawn here. So let's just look at the anatomy of these two amino acids. We've got glycine on the left and alanine on the right. So let's look at the anatomy of glycine. Here's the carbon, here's the alpha carbon in the middle, and here's the hydrogen that hangs off the bottom. And the R group in glycine is just a single H, super easy. So now, this amino acid has had its amino group drawn at physiological pH. At about pH 7.4, this amino group actually acts like a base and it absorbs a proton from solution, and that means that this nitrogen becomes positively charged. So this nitrogen is behaving a little bit oddly. Don't worry about the chemistry of that. You just need to accept that this is also a correct way of drawing this amino acid. And then the carboxylic acid at this pH has acted like an acid, and it's lost a proton and left the carboxylic acid group with a charged oxygen. So these are complete charges on this oxygen and on this nitrogen. So that's an okay way of drawing glycine. And here's alanine. Let's look at its anatomy. There's the alpha carbon. The R group of alanine is CH3, and we've got carboxylic acid and then amino group. Now, it's a little harder to see the condensation chemistry when the amino acids are drawn like this. So I would encourage you, when you're drawing your amino acids, to not actually draw them in this state. These are ionized because they've either gained or lost a proton, depending if we're talking about the amino or carboxy terminus. So I actually, when I do this, if I'm going to draw these to be joined together, I would actually put the H back on here to form a hydroxyl, and I would delete one of these 3 hydrogens, so there's only 2 over here. But this is actually the physiologically correct way to draw these at physiological pH. Now, in the ribosome, which is the machine which is going to join these two things together, the pH is altered a little bit, and the chemistry means that this COO here has actually got a hydrogen down here, so it's COOH, and the NH3 here is NH2. So, so in the ribosome, actually, is what's going to happen is we're going to take the OH from down here. So you can imagine there's a hydrogen down here. We're going to take the OH from here. Then we're going to take one of the 2 hydrogens, and remember that when we take when we take an OH and an H, we're going to get a water molecule, so we're going to get H2O. And so what ends up happening is that this carbon becomes chemically linked to this nitrogen, and that's this red bond down here. So let's just take a look at the resulting molecule down here. Now, this is called a dipeptide because it's got 2 amino acids. So let's take a look at this dipeptide at the bottom and find out where the pieces from glycine and alanine have ended up. So let's start from the left. Here is the amino terminus, or the amino group on the glycine, and here is the amino group in the resulting dipeptide. Here is the alpha carbon of glycine, and here is the alpha carbon in the dipeptide. Here is the C of the carboxy group that we had in glycine, and here it is in the dipeptide. So this double bonded oxygen here is this atom up here in glycine. Then we've got a peptide bond joining us to the nitrogen, so that's the nitrogen of alanine. Here it is. And we lost one of the hydrogens. Remember, I asked you to just pretend there's only 2 hydrogens on here. So if we lose 2 hydrogens, we end up with 1, and there it is. And then we've got the alpha carbon of alanine there, and here it is. Here's our R, our CH3 in alanine, and there it is up here. And then we've got our carboxylic acid group on the end, and bingo, there's our water molecule. So is what you can do, and I'll ask you to do this a little bit later, is actually try and do this yourself. So we'll come back to that in a little bit, just to do a little bit of revision. So is what happens in cells is these amino acids all get linked together in particular orders, which are dictated by the sequences of nucleotides in genes. So your genes determine what sequence of amino acids are linked together to give you a functional protein. So a protein is a long chain of amino acids that's folded up and does a job in your cells. So ribosomes do that linking together that we've talked about before in terms of forming polymers. And so is what we're seeing here is a short chain of amino acids, and the peptide bonds are linked, are shown in red. Excuse me, I need a sip of tea. Throat gets a bit scratchy doing this all day. Okay, so there is our amino terminus from the first amino acid. There's the alpha carbon. There is what's left of the carboxylic acid group. joined to the next amino acid by a peptide bond. Here was the N-terminus, or the amino terminus, or the amino group of that protein. You can see it's largely been destroyed because all that's left is this NH. And then here is the CH3, that's the R group of the next amino acid. Here's the alpha carbon. Here is the C in the carboxylic acid group, and you can see that's all left is this double bonded O. So a couple of slides ago I said that when you look at the chemical properties of an amino acid, you only look at the R group. And so that is because when you link amino acids together and you form a polypeptide, which is a chain of amino acids, you destroy the carboxy group and you destroy the amino group, except in the two amino acids on the end. So the amino acid on the end here has got a free amino group, and the amino acid on the end down here has got a free carboxyl group. So polypeptides and proteins have discrete chemical ends, and every other carboxy group, carboxylic acid group, and amino group in the amino acids that are joined together is basically destroyed, and all that's left are these C double bonded O's, or these NH in the peptide backbone. And so this repeating chain of NCCN, CCN, 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 that's called the polypeptide backbone. And so as you look at that polypeptide backbone, is what you should start to see is that there are some interesting chemical properties in the peptide backbone. So this C double bonded to an O here, the carbon, remember, is not very electronegative. The oxygen is highly electronegative, so the electrons are pulled towards the oxygen in this case. Now when we've got an N and an H next to one another, forming a covalent bond, sharing electrons, the electrons are pulled towards the N, and the H becomes slightly positively charged. So we've got slightly negative oxygen, slightly positive hydrogen, no, it's nonpolar, slightly negative oxygen, slightly positive hydrogen, it's nonpolar, slightly negative oxygen. So what we have are slight negative and positive charges all the way along the peptide backbone. That's going to become really important when we look at protein folding in a moment. One of the ways we can represent these polypeptides is in a little diagram where the amino acids are shown as balls, the peptide bonds are red solid lines, and this NH3 here again is being shown at physiological pH, so it's got an extra proton, and that is called the amino terminus, or the N terminus, N because of the nitrogen that's present there. And down the other end, the carboxylic acid group is the C terminus because of the C in the carboxylic acid group. So I've been talking for a little while. This is going to be a slightly longer discussion than we've had so far. I'm going to pause for a few minutes, and I'd like you to stop the video in a moment, and I'd like you to try and draw a glycine amino acid based on whether you can recall the generic amino acid structure. So draw glycine, and you can do this one of two ways. You can get your book out, and you can sit and you can copy from your book onto a piece of paper, or you can flick back in the video and you can copy from the video onto a piece of paper, or you can actually do what will help your brain learn some of this stuff, and that is to do as much as you can from memory, and then go back and check and fill in the things that you're struggling with. But an important first step is actually getting some stuff wrong, making mistakes, and seeing where you're struggling and what pieces don't you remember. So try and draw glycine, and then when you get done with that, try the second part in grey on the bottom of the slide. So pause the video and we'll be back in a moment. Okay, so we've taken a look at amino acid structure and a couple of examples of amino acid structure, and we've looked to see how you can join some amino acids together to form a polypeptide. So what we're going to do next is look at how polypeptides fold up. So when a polypeptide is formed, it is just a chain of amino acids. That's what ribosomes do. You've heard of ribosomes. They just join amino acids together in a chain, in a sequence which is determined by your genes. So a polypeptide is not the same as a protein. A protein is a fully folded, fully functional chain of amino acids, and they have complex three-dimensional structures because they do complex things in cells. These are things like enzymes and antibodies and stuff like that. So when we think about how a protein folds up, it can go through 4 levels of structure. Now, all proteins are going to have the lowest level, which is called primary structure. And so primary structure is shown over here on the left. That is just a chain of amino acids. So we've got methionine joined to proline, tyrosine, leucine, histidine. Then the dotted line here represents a whole bunch of other amino acids that the artist obviously didn't want to draw out because of space. Then we've got some arginine, proline, tyrosine, leucine, histidine, and then we've got the carboxy terminus. So that's primary sequence. All proteins must have primary sequence because they are chains of amino acids. So then what happens? Well, remember how I explained that you've got the C double bonded to an O in the peptide backbone, and you can zip backwards and look at this and then come back here if you need to. You've got the C double bonded to an O, and the oxygen is electronegative, so it's got a slight negative partial charge. And then you've got the NHs off of the peptide backbone, and because the nitrogen is electronegative, the hydrogen becomes slightly positive. So in a cell, when this chain of amino acids is kind of, it's just wiggling around like a kind of bit of a, a crazy snake, if you like, it's wriggling around, and if a slightly positively charged hydrogen in the peptide backbone comes close to a slightly negatively charged oxygen, hopefully you can guess what happens. You get a hydrogen bond forming, and when you get these hydrogen bond formings between the C double bonded O's and the NH groups on the peptide backbone, you can get two types of structure formed. One is called an alpha helix, which is shown here at the top in purple, and then the other is a beta pleated sheet, which is shown at the bottom here. Now, you don't have to worry about what causes each of these structures to form, but these are the examples of secondary structure. So we always have primary structure. Invariably, you have secondary structure, either alpha helix or beta sheet. And here's the important thing. That is due only to hydrogen bonding between opposite partial charges along the peptide backbone. Now we're getting into the situation where if you try to dismiss all that stuff about electronegativity and what a hydrogen bond is, this doesn't make any sense and it's going to start going off the rails. So this is where we start losing students because they haven't really grasped the idea of electronegativity and hydrogen bonding from that discussion of water. And it seemed pretty stupid to be talking about water in a biology class, but this is why we do it. So if you're struggling, go back to that video, get on board with polarity, electronegativity, and hydrogen bonding. Okay, so that's secondary structure. Off my soapbox now. That's secondary structure. Once you have secondary structures, you can think that they're also they have some free movement. And what will happen is different pieces of secondary structure can stick together, can associate with one another, and that gives us what's called tertiary structure. So tertiary structure is just simply shown here. So tertiary structure is what we've got a piece of alpha helix here associated with some beta pleated sheets, and so you can see that's all kind of wavy beta pleated sheets, and this little alpha helix is easy to see. So those two structures are stuck together, and we'll talk about how the two structures there become stuck together. But you can have extensive regions of alpha helix, extensive regions of beta pleated sheets, multiples of those in a protein, in a folding protein, and so they start associating together. You can get alpha helix, beta sheet, alpha helix, beta sheet, and so these things all start folding into complex three-dimensional structures, and without that structure you don't get function. And you have to have function because your cell is dependent on these proteins for a lot of the doing that happens in cells. So a lot of the work of the enzymes dependent on the three-dimensional structure, their architecture. So that's primary, secondary, and tertiary. Now some proteins have an additional level, and so the additional level is called the quaternary structure. This is confusing for students, and I've not yet kind of found out why it's confusing. If you've got two proteins with tertiary structure, two separate polypeptide chains, if they happen to stick together, if they've got faces which match one another, like two pieces of a jigsaw, and they can be stuck together, that's quaternary structure. So it's two polypeptide chains folded up, maybe just having secondary structure, but usually having tertiary structure. When they stick together, two separate polypeptides sticking together, that is quaternary structure. Hemoglobin is a classic example of this. It's got 4 subunits, 4 individual polypeptides. They all come together, and that whole structure is said to be quaternary structure. And I'll give you some more examples of this as they come up. Okay, so I want to go back to tertiary structure now, because this is a little bit more complicated. Now, Remember the secondary structure, and here comes Eddie. You going to say hello, buddy? Yeah, I see you there. Okay, back to sleep. So, tertiary structure. Secondary structure, that's only dependent on hydrogen bonding. Tertiary structure is dependent on a lot of types of interactions, and here's the key part to this. These interactions are between the R groups. So secondary structure, the hydrogen bonding is between the groups on the peptide backbone. But when we talk about tertiary structure, we're talking about a variety of bonds and associations that form between the R groups. So we've got these R groups kind of hanging off in our alpha helices and beta sheets, and they can associate with one another. So what kind of bonds do we have in tertiary structure? We can have hydrogen bonds. So here's a hydroxyl group. It's got a slightly positively charged hydrogen. If that doesn't make sense, why? You've got to go back and fix that problem. And we've got a electronegative oxygen here, slightly negatively charged, and so we get a hydrogen bond. We can also get what's called a hydrophobic interaction. I don't want you to worry about van der Waals interactions. Chemistry can deal with that with you. Hydrophobic interactions. We already talked about how fatty things stick together and exclude water. So if you've got two amino acids like this one here and this one here with fatty R groups, these are both hydrophobic R groups, they're going to stick together. Disulfide bridges. Now remember I said there were two amino acids, cysteine and methionine, which have sulfur. The reason that becomes important is because if you put two cysteines close together, and the pH is just right and the chemistry is just right, the two sulfurs let go of hydrogens they're associated with, so they act as weak acids, and then the two sulfurs become connected together. So it's what we've got here. This here is the R group of a cysteine, and then this is the R group of another cysteine. And when they come close enough together in two pieces of secondary structure, they can form this really strong disulfide bridge. Lastly, we've got an ionic bond. So an ionic bond is between two complete opposite charges. So here we've got a carboxylic acid in an R group, which has lost a hydrogen, so it's negatively charged now. And we've got an amino group here, which has gained a proton and is now positively charged. So these are complete positive charges. That's very different than in a hydrogen bond, where we're dealing only with partial charges due to differences in electronegativity. Okay, so those are the types of interaction which give you tertiary structure. It's getting complicated now, isn't it, folks? So this is where it starts getting hard to keep up. But this is all based on understanding that early basic chemistry. So what I think you can do to help yourselves along here is create a table that basically compares and contrasts secondary and tertiary levels of protein structure. And you can throw in primary structure there if you want as well. So primary, secondary, and tertiary, 3-column table if you like. Compare secondary and tertiary levels of protein structure. We'll take a pause and start the video up again in a moment. Alrighty, okay, hopefully you did okay with that. If you struggle with that kind of thing and you want some help, throw something in a discussion and see what other people have done. Take a photo of your table with your phone, upload it into a discussion, and have a chat with some students. For that kind of thing, I'm not just going to post the answers to that stuff. The more you discuss that and engage with the content, the more likely you are to learn it. And me just telling you all this stuff doesn't really help you, to be honest. All it does is establishes what you need to know and what you need to understand, and gives you some help with the complicated stuff. Right, let's finish up with some questions here. We've got 4 or 5 questions, I think. So I will leave the question up, pause the video, and then I'll post the answer like I've been doing. So here we go. Here's your questions. Let's see how you get on with this stuff. And here's your second question. And here's your third question. This is the last question coming up now. Okay, so that is a lot of information about proteins. Don't forget to check out your learning outcomes. There'll be some additional things in there. Make sure you do those activities that I put into this video, and I will see you next time when we're going to talk about nucleic acids, and it'll be a little bit shorter, I promise you.